Hi everyone. This video is about proteins, protein structures, and protein functions. You can see in the picture here there's a variety of food sources that we all know are high in proteins. We've got meat in the form of fish and chicken and red meat, eggs, beans, other kinds of nuts and seeds. These are all really excellent sources of proteins. But did you know that proteins have a lot of other functions other than just for making food? Here's a series of pictures that indicate to you some of the functions of proteins. So I want you to just take a moment and look at the pictures and think about what these functions might be. In your note from the Biochem Note 4, there's a space at the top of the protein section for you to brainstorm a list of functions. So you should pause this video right now, look at these pictures, and write down what you think the functions of proteins are in living things. Okay, so I hope you paused and I hope you wrote some ideas down. I'm going to talk to you about what you see here on this page. So we have, of course, muscles. We've got a body from the Body Worlds exhibit. If you haven't seen it, it's amazing. He's kicking a soccer ball and you can see all the muscles of the body. We have skin, and we have a lot of protein in our skin to give it that elasticity. Feathers, hair, and antlers. Structures like feathers, hair, and antlers all contain proteins as well. So these structures that you see, oh, here we go again, I'm moving things around. These structures to the left, these are all structural functions for proteins. The proteins help to create these structures. Okay, they provide that scaffolding for other molecules to create these more complex structures. The two structures or pictures that are on the right of the screen, these are proteins that have more of a functional role within living things. So the top one is a steroid hormone. I think that's estrogen actually. And our steroid hormones are made of protein. Actually, I'm messing that up. Our steroid hormones are made of lipids actually. We've already covered that. So I shouldn't have put this hormone in here, but other hormones are protein. Sorry about that, a little mistake. Most of our hormones are proteins. The steroids are lipids. Probably the most important function for proteins in our bodies are as enzymes. So this picture down here, enzymes are protein catalysts. They are molecules that speed up chemical reactions. They can also slow them down, but most of our enzymes speed up chemical reactions by making the reaction happen more easily. So these are some of the basic functions of proteins. So I'll move on to the harder part of this lesson, which is about the structures of proteins. Proteins are made up of these long chains of repeating molecules. So they are polymers, and the monomer that makes up the polymer, called a protein, is an amino acid. And you've probably all heard of amino acids before. So amino acids are the monomers of the polymer that we would call a protein. There are 20 different amino acids that make up proteins. They all have the same general structure, but one part of each of the 20 different amino acids are a little bit different in terms of their atomic structure. Now for humans, out of the 20 different amino acids, 8 of them are considered essential amino acids, meaning we cannot synthesize them in our body. We can't make them in our cells. We have to get those 8 essential amino acids in our diet. And so that's why it's important to eat a variety of foods. If you become deficient in one of those essential amino acids, you can have a form of malnutrition. So it's important that you eat a variety of foods. It depends on the species. Other species of animals have a different set of essential amino acids. And I know, for example, with cats and dogs, they have their own set of essential amino acids, and that's what the dog food and cat food companies have to make sure that they put into those foods. So this is a graphic of the basic amino acid structure. All of the 20 different amino acids have a central carbon, and around that central carbon there's a hydrogen, and then there's 2 functional groups, one on each side. One of the functional groups is called the amino group, and it has nitrogen in it. So this is the first time that we've seen a macromolecule with nitrogen in it. And the amino group is an NH3, or if it's in an ionized form, it'll be NH2. The other functional group is a carboxyl group, and you saw these with the carbohydrates already and with the fatty acids. So a carboxyl group is a COO or a COOH, depending if it is ionized. So here we have the ionized form with the this O right here. It's lost its hydrogen, so often there's an OH here. And so that's the COOH. Now, what's really interesting about amino acid structure is down here. So this is called the R group. And we use the letter R just to denote that there is something there. There is some chemical structure there. And this is what's different in each of the different 20 amino acids. So they all have the same basic structure that I just talked about, but they each have a different R group. Some of the amino acids are really simple, and their R group is just a hydrogen. Other amino acids are really complex, and they'll have like a set of ring structures in their R group. But that's what sets one amino acid apart from a different one. So this graphic shows you the different amino acids, and it's a little bit small. I couldn't make it any bigger, so I know that's hard to see. But all of the different amino acids are in your textbook, but I just wanted to show this graphic to show you that they are grouped by their chemical properties. So we have in the top here, we have some amino acids that are electrically charged. They're either positive or they're negatively charged, and that is based on what's in their R group. So you can see the R groups are here. They all have the same basic structure, but their R group is differing. Some amino acids have a polar side chain, but it doesn't have a charge. It's just polar because of its shape. Some have a hydrophobic side chain, so they're nonpolar, so they're afraid of water. They're hydrophobic. And then there are a few that have very special R groups that set them apart from everything else. You can't see this very well, I know, but this one right here, cysteine, it's a really important amino acid in protein structure, and the reason is because of in its R group, it has this SH functional group. It's called the sulfhydryl group, and so that's cysteine. Just keep that one in the back of your mind because we'll be coming back to that later. So here's the tough part, but hopefully you're starting to recognize a pattern here. This slide shows you the reaction that happens between two amino acids in order to put them together. So proteins are, remember, hundreds to thousands of amino acids linked together. So it really is pointless to study an amino acid in isolation when in reality a protein is made up of hundreds to thousands of them. So we want to look at how do these amino acids come together and form a bond. So in this graphic I'm showing you two amino acids. They each will have a different R chain or side group, but they both have the amino group and they both have the carboxyl group. They will have a condensation reaction or a dehydration synthesis reaction. And we've seen this with the carbohydrates. We've seen it when a fatty acid attaches to a glycerol to make a triglyceride. So there's really nothing too new here. But you can see that a reaction will happen. Uh-oh, want to draw on this? It's probably not going to let me. Okay, so there will be a reaction between the carboxyl group of one amino acid and the amino group of the other amino acid. And so the reaction is going to be between an OH and an H, and that would happen right here. And of course when we put an H and an OH together, it makes water. So here's your water. Here's the bond between the two amino acids. It's between the nitrogen of one amino acid and the carbon from the carboxyl group of the other amino acid. And with proteins, that bond is called a peptide bond. So far with the carbohydrates you've seen glycosidic bonds. With the lipids you saw ester bonds between fatty acids and glycerol. With the proteins, the bond is called a peptide bond. And I had encouraged you early in the carbohydrate video to make a chart in your notebook where you could start to put this bond information because it's really hard to keep it all straight. When we do nucleic acids next, I'm going to introduce another new type of bond. So just to keep it straight, peptide bonds connect amino acids in protein structure. Anytime you see the word peptide, Think protein. So I'd have to say that proteins have the most complex structure out of all the molecules that we've been studying so far. And the reason is because they really have 4 different levels of structure. From very, very simple, you can break it down to a really simple level of structure, but then those proteins become much more complex as they're made. So proteins are made from your DNA. Your DNA is the code for making proteins. And hopefully you learned this in grade 11, but the code in your DNA tells the ribosomes in your cells what amino acid to put on the chain next. And that sequence of amino acids is called the primary structure of the protein. So let's say there's 1,000 amino acids in a protein. Remember, there are 20 different amino acids. It is the sequence of those different amino acids in that 1,000 long chain of amino acids that determines a protein's function. So we have thousands of different proteins in our body. Each one has its own unique sequence of these 20 different amino acids. And that's referred to as the primary structure. That's the polypeptide. Okay, poly meaning many, peptide means protein, so it's many, many amino acids that creates the protein. And this is referred to as the primary structure. Now, once that chain of amino acids has been made at the ribosome, that chain of molecules will then either coil up or it will bend up. And what I mean is, if you think of a phone cord that's coiled, that's one way that amino acids can form a structure, or they will bend up like taking a piece of paper and folding it into like an accordion shape or a fan shape. And these two structures are called alpha helix, that's the coil, or beta pleated sheet. It depends on the protein, which one it will become. Generally, our enzymes and our hormones and transport proteins that move things in and out of cells or around our body and our blood, they will have alpha helix. They'll be a coiled up chain. If the protein has more of a structural role, like in our muscles, in our skin, in our hair, and our nails, it will form the beta pleated sheet. So that's kind of a generality there. That's referred to as secondary structure. It's alpha helix or it's beta pleated. And there are some intermolecular forces that help to hold the polypeptide in either that alpha helix or that beta pleated sheet. There are some really great graphics in your textbook that you should probably take a look at to help you get a visual of what this looks like. Moving on to the tertiary structure, or the third level of structure, the alpha helix, or the beta pleated sheet, whichever it is, will fold up on itself even further. And we get some really important bonding happening that helps to hold that protein in that shape. Now, what we see is, first of all, disulfide bridges. So remember those cysteines that had the sulfhydryl group? If you get two cysteines close together, they will form a bond. So they will have a reaction and the sulfurs will bond together from the sulfhydryl group. We call them disulfide bridges. So again, that's very important in holding that tertiary level of structure. You also have a lot of hydrogen bonding. When you bring those amino acids close to each other, there's a lot of oxygens, there's a lot of nitrogens, and there's a ton of hydrogen in there. Because remember, all those amino acids have nitrogen, oxygen, and hydrogen in them. So you just need to bring those hydrogens close to nitrogen and oxygen are going to pull on those hydrogens. They're going to pull on that valence electron that hydrogen has. So we see a lot of hydrogen bonding in that tertiary structure as well, and this helps to hold the protein in that shape. One of the things I want you to never forget is that when you change the shape of a protein, it loses its function. It will no longer work. So all of these intermolecular forces holding a protein in its tertiary level of structure and its secondary level are very, very important. If you heat up a protein or you expose it to the wrong pH, all of these intermolecular bonds will break and the protein will fall apart. It'll cease to function. And the last level of structure is called quaternary structure, or the fourth level of structure. This does not always happen. Only some proteins go this far to the quaternary level. And what happens in the quaternary level is that two or more different proteins will come together and form a really big complex. And it's only when they are associating together and they form this big 3D complex does it actually work. So then it's a completed protein. A really common example is hemoglobin. That's the protein in your red blood cells that carries oxygen and carbon dioxide. It's actually made of 4 different polypeptide chains that form alpha helixes, and they come together and form this big complex with an iron atom at the center of the complex, and it's the iron that will bind oxygen. So 4 different levels of structure, starting out really simply with our sequence of amino acids. That's called primary structure. And then it coils up or it bends up into the beta-pleated sheet shape. The tertiary structure, that structure will then fold up on itself. We see disulfide bridges and a lot of hydrogen bonding happening to hold it in that shape. And finally, quaternary structure only happens with certain proteins. So here's a really nice graphic I found to show you those 4 levels of structure. So we have the primary structure here, and you can see it's just the different amino acids. So we have glycine and phenylalanine, glutamic acid, alanine, arginine, aspartic acid, cysteine. It's that sequence of amino acids that forms the primary structure. And that sequence was in your code in your DNA, and your ribosome constructed it in the proper order so that it would take on its shape and have the proper function. The next picture is the secondary structure, and you can see the alpha helix right here. And that's why I say it looks like a phone cord. It's all coiled up. That chain is all coiled up. The other structure it could form is a beta-pleated sheet. It's a little hard to explain this, but imagine taking a bunch of ribbons and laying them beside each other by looping them back and forth, back and forth. That's kind of what beta-pleated sheets look like. The tertiary structure, well, it's hard to explain, but basically your alpha helix and your beta-pleated sheet structures just loop all up on themselves. It's almost like making a ball of yarn, winding it all up, and then you get these forces of attraction between cysteines forming disulfide bridges and a whole lot of hydrogen bonding holding it in that shape. And then finally, quaternary structure. You can see here's a protein. They've kind of modeled it to have 4 different polypeptides all associating together to create that final shape. It's kind of what hemoglobin looks like. And again, only some proteins will enter into a quaternary structure. So that brings me to the end of the protein video. Proteins are complicated, and I went over a lot here, so I hope you take some time to read your textbook and the note over and ask me questions when you don't understand something. The next video will be about nucleic acids, which includes DNA and RNA and a few other interesting molecules that we can't live without in our cells.